Hi, we're continuing with my notes, life science and cell biology in a nutshell. So, um, previously we were learning about um, evolution and how the more similar the DNA of various species are, the more closely related they are. DNA analysis a way is an objective way versus visual observation, which is also used. So, DNA analysis using microscopes, more molecular level, is a method that you can get more specific results about the evolutionary histories of various organisms and the commonalities of their DNA. Um, Also used is the fossil record or structural analysis or visual observation, which is also used. And so these ways are used in order to classify groups of organisms based on their similarities or commonalities of their traits. Homologous structures are structures that are similar across different species. And adaptation is when traits of organisms help them in living in a specific kind of environment. And natural selection is when those organisms with more suitable traits are able to pass on their genetic um, to their offspring. So over time, evolution occurs, and um, the more favorable genes are passed on to offspring. So previously, we learned also about genes, genetics, and how genes are or DNA molecules are turned into proteins via transcription and translation, and mRNA molecules, messenger and transfer RNA molecules helping out with this process. This protein synthesis occurs on a cell organelle called a ribosome. It is a cell structure where proteins are produced. These look like kind of like grains inside the cell cytoplasm. This is showing what this ribosome looks like. It functions as a location for protein synthesis off of mRNA molecules that come out of the nucleus through nuclear pores and latch themselves onto the ribosome in order to make proteins off these mRNA strands with the help of tRNA. Here you have, let's see, the mRNA here, the 3' end, the polypeptide chain growing on the opposite end. tRNA molecules help with this. So the amino acid binding site where the tRNA um, brings the amino acids based on codons on the mRNA and anticodons of the tRNA. And each anticodon brings a different amino acid. So as the amino acids grow, the amino acids bind and become a polypeptide chain. So this is showing, the large ribosomal subunit, the small ribosomal subunit. Okay. So the genetic code results in a specific protein being synthesized, basically. In the translation process in the ribosome, so each subunit of the ribosome is an aggregate of ribosomal RNA molecules and proteins. It has 3 sites binding the 3 types of RNA, transfer RNA, mRNA, etc. This is a 3D structure showing what the ribosome looks like. It's a complex molecule which is made of the 2 major subunits. A cell assesses specific genes by transcribing their DNA sequences into RNA sequences. A cell's characteristics reflect control of transcription. So DNA to RNA is transcription. RNA carrying a genetic information into the cytoplasm enables it to be translated into proteins, amino acid sequence. So the mRNA carries the gene sequence information, and our ribosomal RNA, part of the ribosomes, brings together these mRNA with the amino acids as a protein is formed. tRNA molecules match specific amino acids to specific mRNA triplets called codons, enabling ribosomes to assemble proteins. To synthesize a protein, tRNA molecules carry these amino acids, which pair with complementary anticodons to codons, and from the tRNA to mRNA, ribosomes align the amino acids. Then the protein is folded into certain conformations in order to function the way it's supposed to. A change in the gene's nucleotide sequence can alter the gene's function and affect the phenotype, which is the protein that is the protein will be different. So if you change the way the DNA is expressed, then you will have different proteins being made. And therefore the phenotype or the expressed attributes will be different as well. So these molecules are very big and complex in 3D. So this is how they look like in transcription and translation with the coding strands, etc. So replication from DNA to RNA, and then so DNA coding strand yielding the DNA, the RNA, that's transcription. Replication is just purely doubling the DNA amount. So then the mRNA exits the nucleus, the ribosome. Ribosomal RNA binds with proteins within the cytoplasm to form ribosomes where proteins are made. Transfer occurs with tRNA, which transfers amino acids to the ribosomal units to make the polypeptide chains. This is what tRNA looks like. This is the anticodon binding site, which binds to the codons of the mRNA molecule. So DNA, mRNA, each triple base is a codon, and each one determines which amino acid it's going to go. So for example, if you have adenine, guanine, this codon stands for the amino acid being brought, which is lysine. If you have uracil, cytosine, adenine, the amino acid serine is going there to that codon. If you have guanine, uracil, cytosine, you have valine amino acid. So then you just adding all these amino acids creates a sequence of amino acids, which creates the